We are at the hour. A couple of people are coming, trickling in. I want to say hello. This is a part of our monthly webinar series. Every month we pick a topic area that is really close to our heart, and we invite experts and collaborators. This month is a very special one. We actually have folks from RedShiftBio joining us today. Over the last year and a half, we developed together a pretty comprehensive life science lab ecosystem that includes, not only an all-in-one box, but also a touch interface, and then recently also some accessories like a buffer station. We will get into the details of that. And we actually got a bunch of design awards for it, which is really always a good way to know that you're going in the right direction. Not that we ever design for design awards, but it's always fun to get them. And especially in this one, it's a very cross-functional team that actually collaborated on this. And I want to kick this off by introducing our panelists. First but not, how do you say first but not least? No. David Sloan, actually from RedShiftBio. He's a vice president of life sciences. And David, you want to talk a little bit of background and how you ended up in this space?
Sure. Thanks, Gregor. So I've been with Redshift for about 3.5 years. I've spent most of my biotech career, which is about 20 couple years, working kind of in a customer-facing role and making certain that the products that the companies that I've worked for are meeting customer needs. It's always critical that whatever you're selling or whatever you're supporting solves scientific problems, but it's even more important that it is a product that a customer can work with, that it's something that's going to fit into their workflow, fit into their lab, and make them successful and productive. And I've been in the world of, so Redshift is a biophysical characterization company. I've lived in the biophysical world. I've lived in the proteomic world. I've also done some projects and worked with some companies in the genomics world as well. So I've kind of done a tour of many different areas within the life sciences. And with that, I'll turn it back over to Gregor.
Excellent. Yeah. And I think, I'm very excited, David, to have you on, because you are this critical connection point between the customer's needs and the technology capabilities. And, you talk to customers every day, but at the same time, you also understand the science, which was really helpful when we developed, especially the user experience design and the overall experience of the instrument, to have access to your, first kind of like hear it from the horse's mouth kind of approach. At the same time, also, many UX designers kind of fall into this trap to make something as complex as humanly mankind can do. But you always said, hey, let's keep it simple. Let's really focus on the user needs. And it was really helpful for us, I think, to galvanize the entire team around kind of the mission of what Aurora actually became. Next person on the panel.
Just add one last thing. Well, we love that Loft won design awards for Aurora. We're also really proud that we won a new product award as well for Aurora, not excluding design, but also just for what it does and how it works. It was awarded to us at a different conference at PEGS in Boston not too long back. So super pleased to have won that award as well. So we kind of had both ends of the spectrum covered, with the design award as well as a functionality award and what it's doing for people. So couldn't be happier with how that turned out.
Yeah, and I think, we were, our engineering team was also very heavily involved in kind of the function and kind of like the subsystem development. So yeah, it's definitely a great way to think about it. And I think life sciences, as we are kind of pushing, more advanced science and kind of more complicated processes, instruments that are smart, that really help scientists do better work, are, are becoming more and more prevalent. And it's great to see the industry really moving in that direction, with integrated touch panels and, advanced workflows and advanced sample preps. I want to actually turn over to Valerie, who was really instrumental in also this project. And maybe you can introduce yourself. She's the application manager at RedShiftBio. I'd love to hear about your background and also your affiliation with Redshift.
Yeah, thank you so much. So I've been with Redshift for about 6 years now. I came directly from grad school. I was at Clark University in Worcester, and our group focused on protein structure characterization, mostly using CD and some NMR and fluorescent techniques as well. So we were working with aggregated peptides like amyloid beta, IAPP, insulin, lysozyme, those types of proteins. And we were actually like reached out by my former boss that was like, you guys do a lot of CD work. Would you be interested in testing our new MMS tool? So our group was actually the first beta testers of the first generation instrument from RedShiftBio. And so we ran on a very early system. We ran tons of samples. We loved it. It was so different from CD in so many ways. And so I ended up writing a chapter in my thesis about MMS and then joining the team when I finished at Clark. So I was first beta tester, and then 6 years ago, I joined as an application scientist. And it's been really exciting to see the progression from the first generation instrument to the instrument that we're working on now, because they're so different. And but it's been a really good evolution.
Excellent. Yeah, and I recall spending, many sessions with you in the lab, and you guys are coming back from the field and, presenting your work to some of your clients in India, and machine crashing and all kinds of impromptu work. So yeah, it's been really fun working with you, and I'm glad you're on the panel. And then last but not least, Clotilde Grimault. She's Loft's director of product. And yeah, I'd love to hear from your background and a little bit also how you were involved in this project.
Absolutely. So as the director of product for Loft, my passion is about aggregating systems, especially gloriously complicated systems. How do we make them easier to use, pleasant to use? How do we make them attractive to users, both in terms of functionality and in terms of aesthetics? And so that's my passion, is how do we bring complex things to life? And that includes a lot of disciplines, to be honest. So my background is in mechanical engineering, and I also practiced my data science and data strategy. So I love the integration of the physical and digital world, mostly because consumers don't care. When we experience as individuals and as professionals, we experience systems, there's typically a physical and a digital aspect to it, and we don't care. And you see the younger generations, they're really interchangeably in the physical and the digital world. And so nowadays, product design has transitioned from being, like mechanical, scientific, digital. It's all the same in terms of consumer experience. And so this product design was particularly fascinating, obviously, because of the wonderful team we worked with, Val and Dave, but also because it really embodied a simplification exercise. How do we make things simpler rather than more complex?
Right. And I think that's a common thread in Loft's work, right? I mean, we've been doing science instrumentations for a very long time, starting with the very early 908 device work and, kind of working through the years on how to, I would say, add more tech, but at the same time, reduce the cognitive load, right? So scientists want to get a job done. They don't want to, peck through multiple layers of panels on an interface. And especially as you're kind of seeing labs getting more and more crowded, there's a lot of automation happening. But at the same time, each instrument has to really, take as little space as possible. So by integrating touch panels, by integrating kind of like, I remember the first Redshift product, David, maybe you want to talk about this a little bit, was, 3 boxes next to each other. It took a lot of space on the bench. And then we were able, to really miniaturize that. David, do you want to talk a little bit about like the genesis of Aurora, the product we worked on, and how it kind of came out of the first generation products?
Sure, happy to start there. What Gregor's alluding to is many new product development, many projects, they start off with perhaps an internal product name or internal code name, and sometimes they're really secret, sometimes they're really extravagant. To be honest, ours was pretty simple. We called it Mini. Hey, there's pictures. We called it Mini. The whole goal of the project was to make things smaller. As Gregor said, our first generation product was large, it was big. There was a main box, there was a secondary box, there was a computer, there was a monitor. There were just a lot of things that all went into it. And we really realized that our customers didn't want a massive system that took up a whole bench. So one of our goals was to make it smaller, hence the name Mini, but it really went farther than that. We also wanted to minimize the amount of sample that was required. So we wanted to shrink the amount of sample or the volume that people used. And we got more than a 10-fold reduction in sample. So we were really happy with that. But we wanted to reduce or minimize the complexity as well. There used to be a big training burden, and Val maybe will talk about the training, but there used to be a whole lot more that went into getting people comfortable using the system and degassing samples and putting them in special plates and getting the plates and putting it in this little well-designed but carrier that they had to go into the instrument. There was just a lot that it took to make sure people were doing it right. And the idea of Mini kind of brought all that together, that we wanted it smaller, we wanted it simpler, we wanted less sample. We really wanted to make less of an impact on the day-to-day world of the scientist. But on top of it all, we needed to make sure that we were delivering the same high quality data, the same high quality results that those folks had come to expect. It also was perhaps slightly ironic that in the end, as Gregor said, we went with the name Aurora, which isn't small. Aurora's, it's a large, event that covers the sky in the northern hemisphere. But Mini was what it was all about. It was about making it smaller, making it more compact and concise.
Right, and I think one lesson to be learned, if anybody on this call is in the science development space or in the laboratory science development space, is also that we had a very tight deadline. But with that tight deadline, actually, the first quarter, the first 3-4 months of development, we're very open. We just needed to solve some key issues around sample size, speed, but also like width of the instrument. And there was a lot of kind of parallel developments of the Redshift internal team, but also the Loft engineering team on how can we achieve that. And as David said, a lot of times these proteins can be as expensive as an instrument, right? If you can reduce the size of that protein, the sample size of that protein, you open up a completely different market. And I'd love to have Val maybe talk a little bit about like what a small sample size actually gave Aurora the permission to play in.
Yeah, definitely. So kind of early on when we were demoing and talking to customers about our instrument and what it can do, the data that you can get, everybody's really excited about the data itself and the structure level of characterization that you can get. But then that was one of the really big hurdles, was as soon as they say, this is awesome, we want to send some samples, like get an idea of what our samples would look like and what kind of data we can get. That was one of the big hurdles, was asking for a milliliter of sample, which is huge. And so that shut a lot of doors from the perspective of just reaching out to how many people actually had that much sample to send us, to then see what kind of data they could get and what kind of progress they could make. So we still did get a bunch of samples, and we do have enthusiasts that have enough sample that can do that. But then miniaturizing the amount of sample got us into like R&D labs and just many more labs. So getting to work with more scientists, different applications groups, all of that kind of opened up, because now it's so much easier to get the amount of sample that we need to analyze.
Mhm. And I think, early on, Val, when you guys worked on the first platform, also you did a lot of, I would call it kind of almost like consulting work for some of these companies, right? To prove that the technology could work, but also to give them really interesting data insights, right? Can you talk a little bit about also how that informed your roadmap for development? Was it able to kind of like understand that there's new markets or different markets through kind of the work we've done there?
Yeah, for sure. So we have, in addition to selling instruments, we have a structure lab, which you can send samples and get the data from our headquarters here at this demo lab. We also have another demo lab in New York and Oxford. And so working with all these different groups means that we learned a lot about like IND filing, how the data is going to be used by customers and what is valuable to them. So with IND filings, there is a portion where you need to know the higher order structure of your biologic. And so historically, folks have used like either FTIR or CD for that component of IND filing. And so this is one place where we fit in really well as being a higher order structure characterization tool that can be part of that really seamlessly. And so a lot of the early development work was to say that our structure characterization is just as good, if not better, than FTR and CD, what people are used to looking at. But then definitely talking to people that aren't used to looking at secondary structure, showing them what kind of information you can get in addition to their typical workflows. That's kind of like one of the pieces is if people are already looking at structure, then they know that higher order structure matters and that information is useful. But to a group that doesn't usually look at structure, saying like, this is kind of added information that can help out in your workflow in different ways than what you're used to.
I mean, if I may add one thing where kind of scientists and designers work together is all around the workflow, right? Because the kind of what happens before the instrument and what they do after. And that's something that service design is really helpful with, is kind of like, how do we take into consideration all of the experience of the scientist, not just pushing the buttons on the machine, but where is it coming from? How can we simplify the whole experience holistically rather than just replicates what the box is going to do.
Yeah, and I think I don't know if you guys can put the slide back up with Aurora, because I would love to talk a little bit about some of the UX choices or like interface choices we took. If we, one of the key takeaways for me was this light ring, right? So early on, the light ring wasn't there. We just had a touch panel, and Loft is very familiar with kind of like, I would say these like this development efforts in integrating touch panels into an instrument and what that entails, even with like optical bonding and making sure that we kind of get the tightest integration. But at this point, actually, we needed a door, so we not only had to integrate a touch panel into the front glass, we also had to cut a hole in it, right? So there was a lot of effort around this, like access to the instrument. So the way this works for folks that are not familiar is like you take a well plate, you fill it with your samples, you open the door, it's a sliding door, and then you can put the well plate into the instrument, and then you close the door. The instrument then follows a protocol that you preset to kind of run through as many samples as you have on your well plate. But I don't know if it was you, David, or Dennis, but at one point we said, hey, we could maybe have some sort of like ambient light that could indicate, hey, I'm ready to take the sample, or I'm busy right now. So this idea of this light ring actually became pretty quickly kind of like a centerpiece of our design exercise. And I know that when we introduced the instrument, the highlight at the trade show was, some folks might have called it the disco light, but I'd love to hear from David, like first trade show when you showed up with the model and it had the spinning lights. How was that perceived?
Yeah, I'm not going to take credit for being the origin story for the light ring, but the light ring is really popular on many levels. And Greg, I'll go to the trade show in a moment. Gregor didn't mention it, but if you look at the picture all the way on the left, you'll see that there's a little green light at the bottom, and then the rest is blue. So the light ring is actually integrated to bring the user's attention to what they're supposed to be doing or where they're supposed to be interacting with the instrument. So as the little guys, I don't know, the floating plate there indicates, is that's the time when you're supposed to be loading your plate into the instrument. And there's a green light, and that the little grayish door is a garage door. It just slides straight up from the bottom and the plate goes in there. But the green light's meant to kind of bring your eye to what you're supposed to be doing or where you're interacting with the instrument. And yes, the light was very popular, and the sales team was really excited about the disco mode that Gregor referred to, because it's frequently the brightest thing in the room. It definitely is flashy. It definitely draws one's attention to the instrument, and it brings, for those people who go to trade shows, one of the critical things of a trade show is getting people to come to your booth, and it's also giving people something to talk about so that they feel like they have an opening line when they speak to you. And the disco lights are absolutely that. People walk up to you and say, what's up with the lights, or why is it doing that, or do you know the lights are flashing manically? That's my favorite comment that people make. Yes, I'm well aware the lights are flashing manically. I've been here for 6 hours, but the light is both eye-catching, but also really a well-integrated part of the design. It's really a nice feature of bringing it all together. And I think it does somewhat highlight kind of the really nice back and forth interaction that we had with Loft. Because Loft is like, hey, we can do this. What do you think about that? And then somebody was like, yes, we should do that, but why? And then it was, oh, we can do this, we can do that. It can be integrated as all part of it. It kind of serves as a timer for letting you know how long things are going. It's really a nice part of the whole project. And Val, if you wanted to comment when you guys go out there and do trainings for customers, because it's a bright part.
It really is. And so one of my favorite features is the one that you were just talking about, the timer. So it acts as a loading bar while you're running your plate. So with the 96 well plate, it can be going for hours at a time. And from across the lab, you can just look quickly to see, oh, is my plate like halfway done, based on how the lights look, because it behaves as that loading bar where it'll be that light blue color for the whole thing, and then it'll be a different color for how much the layout has progressed since you started. So that's another feature that from training folks have really liked. And in the beginning it was like, oh, is our light broken, because like this much is a different color than the rest? And we were like, no, that's a loading bar. And so when people learn that, they're really excited to see, like, you can see it across the lab, and you can just get a quick glimpse of how far along you are in your protocol.
Yeah, and I think that's a really key insight, right? Because labs, you do a lot of running back and forth to see if your samples are done. If you can kind of just glance across the street and see what's going on, it's a really valuable insight. And, it really helps you in your efficiency in your lab work. I want to talk a little bit about kind of how labs are changing. I know that, we talked about it being crammed. We talked about it being kind of like people are doing more and more work and more advanced processes. And maybe I'm teeing up kind of the TX conversation, but Aurora came out, David, a year ago, roughly, right? Initially.
Yep, it was July of last year, July of 2023.
Yeah. And then quickly we actually kind of through the feedback you guys got, or even before that, through your feedback, realized there were probably some new opportunities for new markets that Aurora could cover. So we quickly actually followed up with an accessory, which is called the buffer station, but also then, incorporated thermal ramping and other features. David, you want to talk about maybe how kind of Aurora as a platform is evolving, and how you kind of thought it was really interesting that the small sample size then actually opened up other opportunities?
First off, because sometimes we use initials, we decided just calling it buffer station were unfortunate initials to go with. So it's the external buffer station. It's we had to be a little careful with naming. So it's the EBS, please, the external buffer station. But yes, we added an external buffer station because it does increase the throughput. Val mentioned it takes a little bit of time. You can't rush good data, but it was really important to give customers the ability to put as many samples as they wanted on the plate. And you can load up a full plate, and really the beauty of it is you can do it, let's say 2 or 3 or 4 o'clock in the afternoon, hit the start button, and then come back to about 90 fully analyzed samples the next morning. It just gives you a lot of flexibility to run a whole lot of samples with zero interaction with the instrument. You're able just to set it and forget it and go home, and you don't need to be there. So the external buffer station was really a very nice usability and throughput advantage that we had for the system that kind of came out not too long after, and we'll actually say it's startlingly soon after the original system launched, but it's an upgrade that you can put onto the existing system. And Gregor also mentioned it. We did launch kind of a second version of Aurora. We call it Aurora TX. There's 2 really critical attributes that come with TX. One is it makes the system really well suited for the analysis of RNA therapeutics. We somewhat expanded. We didn't get into technology. We don't need to get into technology. Let's suffice it to say that our technology is an infrared spectroscopy instrument that we're working with. And what we did is with Aurora TX is we have a slightly wider spectral range so that we're now really well suited for analyzing RNA therapeutics, so you can understand what is going into your vaccine, what is going into your gene therapy, what does your RNA look like, and how effective is it going to be, and how similar is it to the last batch and the batch before that, and that first golden batch that you created that everything is getting compared to. We also added in what we refer to as thermal ramping or thermal stress, so you can do stability studies, accelerated stability studies to understand, is your drug, is your therapeutic stable? Will it make it through the approval process and be a drug that's going to work for people in real world settings, in real world storage conditions?
Excellent. Yeah. I mean, I think it's interesting, right? We are at this shift in the lab also from kind of new emerging technologies. Before we jump into that, I'd love to also talk a little bit about kind of when we started working on the UX. I think there was a lot of work, and maybe that's a question for Clotilde, around simplification, right? You want to talk a little bit about like the user environment and the user groups and how we kind of weeded through many data sets to kind of ultimately come to a pretty simple workflow?
Absolutely. And so to me, the whole UX development, it's so critical to understand the user workflow first. And quite often we have several users who have different workflow, whether it's an administrator or a user. And so for us, it was all about understanding the best features that they might want on the instrument. And Val, maybe we can talk also at the end about, or a little bit later about why you separated the measurement from the analysis. But for us, it was really all about thinking about the features that are really key to the experience and how do we deliver them seamlessly. And maybe one thing that we had to think about is at some point we have a 96 well plates on the instrument, and we need colors to differentiate all of these things. So there was actually a big color study to make all the colors different given everybody's kind of perception of colors. And so for us, it was all about simplifying. What do we need to show right at that moment? How do we take the step by step while keeping the flexibility and designing with the touchscreen in mind, having to design a keyboard for, when we need a keyboard. And so it was really about simplifying the experience and the visual design of it all.
Right. I think one thing, Clotilde, I'm glad you're bringing this up. One of the things we always hear when we're working, especially in life sciences or in laboratory sciences or in medtech, is that it's really, I would say, crucial to work with an expert like Lav, because we we have filters for color blindness. We understand, human factors principles. We kind of uncover certain things that a lot of times like a software development team just overlooks. And we've seen many UXs where, colors are chosen for marketing reasons, but not necessarily for functionality reasons. And it's really a challenge sometimes to kind of unwind things that just make it into the product. And it was really refreshing to work with the Redshift team, that you guys had such an open mind of saying, hey, let's accomplish this, and how can we get there? And how can we split? We know we needed these functionalities, but how can we kind of restructure our workflow to fit as low of a cognitive load, understanding the human factors principles. At the same time, these labs are sometimes very bright, and sometimes people are tall or short, or it's all kinds of different users that are kind of interacting with this instrument. Also, different user types, right? There's the people that just run samples. There's the lab manager that has to kind of like, give access and permissions. And there's the kind of even the Redshift tech sometimes that has to like remote into the instrument to kind of look something up. I'd love to hear a little bit of from you guys, maybe Val or Clotilde or David, on like, how did you bring in all these stakeholders, right, from a lab to make sure that we have a comprehensive insight from all different perspectives?
Yes, I can start. And I think this really does go back to, we had a first generation instrument that, as Dave was talking about at the beginning, has a monitor, a keyboard, a mouse, all of those kind of accessories that make using a computer really easy. But then going to a touch screen, now all of a sudden typing and having a lot of mouse clicks becomes harder. So the idea here was simplifying, and because we had the first generation instrument to go off of, we had our workflow already kind of nailed down for what we're used to doing. And from that perspective, from a customer, what we used to have them do is fill out, like, you could still do everything automated. You filled out your protocol. You had to select what operations you wanted to do and when. And what we learned from a training aspect is that very often customers would like either putting it all on the customer to like do the wash steps, or different workflows have different steps in between. Having that all on the customer is a lot to ask for. And so in the newer generation, when we were designing this with less button clicks because of the touch screen, we also wanted to simplify for workflow itself and say, if you're a formulation group, we kind of know you're looking at samples all in different buffers. So we can incorporate the proper wash steps and the in-between steps that don't really matter to a customer, but do matter to an instrument health and well-being. So we implemented these sample groups that make it easy for the customer to say, like, this is what I'm doing. I care about my samples. I don't really care about the washing steps that happen in between. So we took care of that for them. And so that minimized the number of mouse clicks, and it minimized the amount that was put on, the burden that was put on the customer to make sure that it was washed properly in between.
I really want to highlight the breakthrough that the sample groups was in terms of UX design, because as we know, we had 96 samples, or I mean, depending on how but we had a large number of samples, and we wanted to minimize user actions. And so we said, okay, all of these samples are going to be of the same pattern. And so we were able to just define, this is the pattern and this is how it's going to change, rather than specifying each and every sample. And I think that was a real breakthrough in terms of usability, making groups of samples that behave the same, where you only have to input it, the input parameters, as minimally as you needed.
As far as working with Loft, one of the things that I really appreciated is they always have kind of a preliminary discovery call where they kind of try to understand what needs to be included in whatever that particular facet has to be. And Chloe is there taking notes, and these little sticky notes, they're all over the digital sticky notes, but the little sticky notes are all over the place, and she's capturing all the information. And then somehow it all gets assimilated, and that's usually at the beginning of the week. And then at the end of the week, there's a follow-up meeting to make sure that kind of everything has been pulled together, and somehow the chaos has been ordered together in a logical flow of how things are gonna happen. And sometimes Val and I had some feedback of maybe we don't like that, but on at least 3 or 4 occasions, I really learned it's not about being right, it's about getting it right. And Loft has a whole lot of experience in, okay, that's how you did it before, but maybe this is a wiser way to do it here. And it really is great to have expertise that comes from the position of having built other, as far as I'm aware, life science instrumentation. I'm sure they built other things too, but other life science instrumentation to really understand the lab and understand the environment. And yes, the front door has to open, but let's make it open this way because it's not going to be as intrusive in the lab, or not going to mess up the instrumentation that's next to it. And the service engineers are really going to appreciate that they're not going to have to be standing on a chair to get in the top, or doing some sort of a contortionist type of position to get around to the back of it. It's all very easily accessible. And it was really a very interesting way of kind of having a back and forth iterative design process to make sure that the instrument was buildable, but also meeting the needs of our customers, meeting the needs of the scientists in the lab.
Yeah, I think, David, also what was wonderful about our collaboration is, we've done a lot of life science and medtech instruments. However, each, subcategory has a completely different workflow, right? So they all are in the lab, but they're all completely work different. So with you and Val being on the team, it was really easy for us to kind of get up to speed on, not everybody has buffer station or buffer requirements or water requirements in between samples, and all these kind of specialty steps that you guys are introducing. It was really wonderful. I think one of the key we love this collaboration, and we love what came out of it, but also I think it also taught us a lot about the limitations of technology and how to overcome it with design choices, right? So the idea of even the external buffer was that was really derived from the fact that you needed a buffer, right? And you needed to kind of give up half of your well plates to do so. So it's all kind of rooted in like how to harness, how to take technology and make it as easy as possible, but then not get too complicated in the overall technical systems to kind of address some of these needs. Clotilde, maybe you could talk a little bit about like the technological side of Redshift, because I think it was also a really good collaboration with Dennis, who's not on the call, but who was kind of leading he's the Chief Technology Officer for Redshift, kind of like, how we were able to, kind of collaborate with the team there. And then I think my next question will be around kind of team size and kind of like how we all work together.
Yeah, so I think as kind of the holistic design professionals around the scientific team, it's always a learning experience. And for me, the magic in the collaboration is in the fact that we had the opportunity to think about the workflow first, the cartridge. We had a whole exploration. What is the best way to bring the samples for the minimal volume at first? And so we started learning about your application. Now buffer and water, buffer, water, buffer, whatever the 4 steps became kind of part of our understanding of the system. So when we were designing the physical experience, we knew kind of what needed to happen. And then when we were designing the digital experience, like, okay, no problem, this is what you need to do. So to me, the long-term collaboration is extremely valuable, so we can really understand what happens scientifically and then support it with design solutions. Another great example, Gregor, that you were mentioning of the design as help for technology. To me, the progress bar is also another solution. We know it's going to take some time. This is kind of like where the technology is. So how can we find a design solution that just makes it pleasant, attractive, right, useful?
Yeah. And then, Chad, who's heading our engineering team, was then saying, we can use addressable LEDs. They're not expensive. And so we instantly had a solution kind of based on that need, but also then that need was also, bringing other functionality like user guidance, but also kind of ambient progress bars over time, but also, you know, trade show bling.
Another thing that I want to bring up in terms of technology integration is the touchscreen. So we knew from day one we wanted the touchscreen, but because of the physical design track, we had to know way before we knew we could design the UX, we had to decide on the size and the configuration of the touchscreen. And so for us, it was really beneficial that we were able to kind of have a long lens view of what was coming next when we're doing the industrial design. Okay, so we know it's going to have to be a rather large screen because you came from a computer and people have gloves. It's going to be kind of this kind of 3D configuration of things. And so being able to decide on the size of the screen before we know anything about the workflow of the UX was kind of like that tension of creativity and technology. How do we find the best balance that's kind of makes it possible for the future steps in the design process?
Right. Yeah. And I'd love to kind of, yeah, maybe David, I think, you know, people, this is was a pretty big endeavor. We did it in, I would say, less than a year, right? From start to finish. Maybe you can talk a little bit about the team size and also the different team disciplines that work together on this.
Sure. Yeah. So Redshift, we're about a 30-person company. We're not the biggest company out there. And this project, we kind of had one or occasionally two representatives from the critical teams that were participating in it. There were a handful of engineers who had to be there so that they could give the engineering input to Chad and the Loft engineering team to make sure everything was going to fit and how it would fit. And I remember large discussions about how big something had to be or could be. And at least on more than one occasion, Gregor said, well, can we tip that 90 degrees? Can we rotate it? Can it be backwards? Does somebody need to have access to it? There was the discussion of, you know, should there be a USB port in the front? And absolutely, yes, there should be a USB port in the front. Our previous instrument was in the back. So why not put it in the front? Let people get to it from the front. But from a Redshift perspective, it was about 7 or 8 people who were really participating in the calls with Loft to make sure that it was happening and that Val and her team were giving the customer input. The software team was there to make sure that the critical features of the software were going to be present, but that it was going to work in that touchscreen environment. And Clotilde mentioned it, but it was really critical, and Loft did a superb job of hammering home the point that someone doesn't want to type in a 20-digit something on a touch screen. A 20-digit something is too long. You'll build great shoulders when you're up there doing it, but it's too long to be typing. So it was the idea of, why don't we pre-populate names to save people the typing? And if they want to edit them, have at it, but don't make them edit it right there on the screen. Give them some sort of a default that you can at least use for exportation purposes, because it was mentioned we now do offline data analysis, and why not change the names there? Or if you want to put the names somewhere else and import them back into the instrument, let that happen. So it was really a great way to, have, a 40-minute meeting once or twice a week, depending on what phase of the project, to make sure that all of the critical people were there so that decisions could be made, so that we could move forward in what was absolutely a very compressed timeline to make everything work, to get the hardware, get the UX, get the software, get all of the workflows together to make for what turned out to be a really great project, a really great product, a really great package that does address a lot of concerns and challenges we had with our first generation system.
David, do you think it really helped that we had the Gen 1?
Yes, it did. But at least for me, what the Gen 1 really does is it really highlights some of the positives we had with our first generation system, but really where we needed to go with that system to make it a great addition to the lab, to really make it a perfect fit for what people were trying to do. We have some customers who had Gen 1 and who have now purchased the Aurora system. We've had some customers who are owners come to a conference or a trade show and see the Generation 2 system at the conference, see Aurora at the conference, and they're really amazed at how we're able to get the same data quality, very similar interface, just so much more streamlined, so much more smooth and user-friendly, and the small sample volume, we refer to Aurora as a one-drop solution because it takes 50 microliters. So it's a very small sample size that is required on this new generation system. So really the comparison between Generation 1 and Generation 2 really gives everybody an appreciation for all the work that was going on behind the scenes to to push out this next generation product.
Right. And I think if I learned something, it was also kind of like a consolidated effort of, Dennis and his team, kind of really honing in on how can we reduce volume, right? Each fitting, how can we get as little fittings as possible? How can we cut down on tube length? And us then actually kind of looking at, how can we kind of package this all up, right? How can we make it so that people can get their fingers in and replace the part and like the human side. So it's really critical to kind of have both of the teams kind of collaborate and understand also what is the critical functionality, right? Like we want to be at as small of a sample volume as possible. We want to be at a particular kind of complexity and price point. We want to be at a particular size. It was really helpful, I think, for everyone. That was pretty clear that what was the goal of the exercise.
Absolutely. I'll also mention the team is somewhat distributed. I happen to be a remote employee, so sometimes I was at headquarters for the meetings. Sometimes I was a remote face and voice. Val brought in some of the field applications team as well, and sometimes they were remote. So we were bringing in the important people, and Loft had their people from different offices as well, attending the meetings and kind of making sure we had the correct bodies together to make decisions so that we could push it forward in the timeline that was required. Because we had to set the size of the screen early on. We also had a launch date early on in the project, and we had to hit our launch date because I also wear a marketing hat. And from my marketing perspective, there was a date, and there was a date on the calendar. We were launching on that date, and we hit it. We hit it fabulously. And
I think it's also, after COVID, supply chains were kind of still crazy, and they still are crazy, but they were even crazier, right? So I recall the touch panel had like a 23-week lead time. So we needed to really galvanize, pick a touch panel very quickly as all the other parts are still moving, so we could get enough for like an initial run. I know we have about 13 minutes left. I'd love to have a last topic and then maybe questions and answers from people on the panel. But my last question really was around, we know that the life science or biotechnology space was a little bit oppressed in the last couple of years after COVID. I would say there was a lot of spending through COVID, and then people are more careful in investing. So you guys shipped an instrument that had like advanced features and new features, quite at the tail of that kind of really shift in the industry. And also, conventional life science processes are somewhat established. I'd love to hear a little bit about kind of the emerging fields and how like Aurora is really fitting in that, and where you see kind of the roadmap for the development. Maybe start with David.
Sure. I was going to call on Val, but sure. Okay.
Well,
either way, I'll hand it off to Val in a moment. What I was really going to say is, from my perspective, I completely agree. There's what's referred to as a COVID hangover. A lot of companies spent a lot of money during COVID because there were really specific set of needs that they had to fulfill during that time. And then some of the companies had decreased revenues afterwards for one reason or another. But from the perspective of introducing a new instrument, introducing a new technology, introducing a new system, it's really about finding the market niche, finding the customers who value that information, who require that information, and who really can't get that information either easily or any other way. And we really feel like Aurora and Aurora TX and the external buffer station solve a lot of those problems for customers, which is why people are still talking to us, which is why people are ordering our instrumentation, which is why our systems are kind of getting prioritized above other products on the market. It is because we are solving problems that they need answers to. I'll hand it off to Val with maybe a lead in by saying, people submit data to regulators, and it's really critical that that data be reproducible and high quality. Val?
Yeah, thanks. So I think we as a company started with protein secondary structure characterization, and with biologics being mostly like monoclonal antibodies. That's kind of where we started. COVID kind of highlighted the possibilities of RNA drugs. And so now where we see the future of these biologics is in characterization of RNA biologics as well. And so with all of the history of protein biologics, we have critical quality attributes, or CQAs, that are set up by the FDA that have all kinds of regulations around it, and all kinds of specifications that folks need to be able to maintain to release a drug. With RNA being such a new type of drug, all of these are being established at the same time. So tools are also having to kind of step it up and say, like, how can we help characterize the RNA products that are coming out? And so I think this is one of the exciting new fields, is that we're all kind of learning together how to characterize and how to best make these different RNA products. And they're all different modalities. We now deal with RNA, but also LNPs, lipid nanoparticles, because that's how they transport. We've branched out to AAVs. So these are all like different modalities that our tool is really well equipped for, but we are learning how to characterize as well. But yeah, so it's been exciting to learn all these new things.
Excellent. Yeah. And I think especially with RNA being kind of, I would say the new, the new chapter in drug discovery, or one of the new chapters of drug discovery. How do you see kind of labs reacting to that? Is there a lot of capital investment? Is it more bigger labs, or is it also institutional, research facilities that kind of really focus on that area?
There, a lot of the investment in new companies, or a lot of the funding from venture capital or from other sources, is going into RNA-based therapeutics, just because the COVID pandemic really highlighted the power of we'll call it the RNA world, and how quickly you can develop new drugs, how quickly you can respond to a need. So both big and small companies are investing quite readily in technologies that can solve problems in the RNA world. It, maybe somewhat jokingly, internally, we kind of refer to it as the Wild West, just because with protein therapeutics that have been around for 20 years, a lot of the tools are very well understood. A lot of the tools are very well characterized, but all those needs haven't quite been mapped out in the RNA world. So the fact that we have a tool that has been around for a while and is well accepted, and not everybody, but scientists understand IR spectroscopy, it's really, we get a very positive reception from the people that we're talking to when we explain to them what we're able to do in the RNA world with this well-established tool that either they were already using, or they have friends and family that are using to characterize protein drugs and do other types of work. So it's not necessary, it's never easy to sell something, but it is, we do solve problems. We are answering questions that people need answered in the RNA world. And it's really very gratifying from a speaking with customers perspective to kind of see that glint of recognition and that excitement about what we're able to do for them and with them with their novel therapeutic modalities.
Mhm. I think we have about 6 minutes left. I want to open it up to our participants, and maybe if you have any questions, please drop them in the chat, or and we can ask our experts. While we're waiting for questions to trickle in, also David, I know anybody who's interested in the instrument can meet you guys in person next week. Maybe you can talk a little bit about what's coming up.
Sure thing. Next week in the Boston area is the Bioprocessing Summit. Redshift will be at the Bioprocessing Summit. Redshift does have two different product lines. We've got the product that we worked on with Loft that's called Aurora and Aurora TX. We have another product that's called Halcon that does a little bit more work in the bioprocessing world. We will have a talk, we will have a poster that's based on that other product line, but Aurora will be on display in our booth. We do have enough people who work in the bioprocessing world on our Aurora technology that will be showcasing it there as well. The conference is 4 days. It's Monday evening through Thursday. I will be there Monday evening and Tuesday. I'm not going to be there Wednesday and Thursday, but the booth is still there. So there'll be other people from Redshift who are personing the booth on those days. So if anybody is attending the conference, please stop by booth 615. We've got fun little marketing giveaways. We're happy to talk to you about our products. We're happy to talk to you about the experience. There, Chloe has one of our fabulous giveaways that that she gestured with right there. But yes, we're happy to talk to people about what we did with Loft, product development, how excited we are about our products that we're out there selling and supporting.
Excellent. Yeah, Seya had a question, and it's basically she's in the stem cell research, and she was asking, basically in a highly regulated system, how can design and technology enhance productivity? It's probably a question for Clotilde.
And also for Val, I would say, because
Yeah. Oh yeah, both of you. Yeah.
yeah. So, Seya, did you want to narrate your question?
Sure. So I have kind of vast experience in, I don't know, every aspect of the pharmacological wheel over the last part of my career, from research and development to electrophysiology to stem cell research, breast cancer research, HIV research, right? So in the laboratory space, as I'm looking now, I'm a project manager for a pharmaceutical company in drug development. And while that is fulfilling, I am realizing that with that vast part of experience, I would like to transition into trying to use those skills in other industries. So what I notice a lot in you all's conversations is some of those things of me having those hands-on skills in different BSL environments could be very useful. So how could I start to use that expertise for companies like you all, pretty much? Could you hear me?
Yes.
Yeah.
Okay, I just want to make sure. It was like a long pause.
Pretty complicated question. We just
Yeah,
maybe
sorry.
Val. I think Val, that's probably a good one for you to get started, and then maybe Clotilde, you can follow up.
Yes.
Sure. Yeah, I'll start. Thank you so much. I think that's a really interesting question. And it does sort of go from like where I came from in grad school, going into an instrumentation company, because I went from user to application scientist. And so having that like user perspective is really helpful, because sometimes in teams that are working on an instrument, you're so tunneled into like what you're working on and how you've done it in the past, or how you like know to do it. But having the experience of working with other tools in your lab, or working with other groups, and like knowing how you can like use it together is really useful in a team like this, where you bring a different point of view and a different perspective
Right.
to how somebody would design a next generation instrument.
Right. Okay, cool.
So that my perspective is that innovation comes from wherever in a way. And so in a way, having multidisciplinary people or multidisciplinary teams is the best way to kind of come up with something new, original, and helpful. And so to at least that's also my background is a little bit all over the place, but in a way, sometimes that allows you to understand different disciplines, different needs, and to then blend them into an innovative package. So it's about kind of leveraging your skills in the right place and time to create something new. Again, innovation is just a bunch of people kind of scratching their head and kind of taking a pencil or a sticky. Dave.
Are you where are you located?
I'm in Raleigh, North Carolina, so I went to
yeah. So we actually quite frequently run like health tech mixers in the area, about 3-4 times a year. I actually included both me and my and Clotilde's email. You can just reach out. We can put you on the mailing list, or just sign up on Loft mailing list. There's lots of stuff happening. I think one of the challenges with, like, I would say a career transition is that if you apply to a job, nobody's going to hire you because they don't know your unique skill sets. So you have to kind of almost network your way into like talking about your skills and then being able to connect to the right folks. So it might take a little bit of a time to kind of connect to the right people, but there's lots of stuff happening in Raleigh in the life science space. I mean, we have an office there. David is there. Clotilde is there. We work with many companies that actually are there. I think it's just getting exposure to the industry. Yeah. Saloni dropped something.
And
and Saloni dropped also like LinkedIn. You can follow all of us on it. We're at time, so I want to actually, Saloni, can you put the last slide up? One more, right? Yeah. Thank you for everybody to join today. It was a really interesting conversation. I love kind of opening up the hood and really, giving some case studies around how we work. Hopefully we can do this more in the future. Next month, we're talking about designing brands that succeed. We have actually 2 folks from the West Coast, Fabian and Adam, and then Jeff from the East Coast joining us. And I'm going to be the host again, but please sign up for that. There's a link in the Eventbrite if you're interested. And everybody, thanks for taking an hour out of your day to join us today. And we're looking forward to keeping in touch. I'll talk to you guys soon. Take care.
Thank you.
Bye-bye.
Thank you. Bye.